Hello, welcome to American Diagnostica Sekisui. Today I will present to you some information on a special research reagent provided by American Diagnostica Sekisui, which is a monoclonal antibody called M3b, and this antibody is specific for the so-called Gla-domains of proteins. In this presentation, I will provide some background information on gamma carboxylation of proteins and the so-called Gla-domains. I will provide some information on the background of monoclonal antibody M3b, which is specific for Gla-domains. I will present the ADI products based on this antibody and some scientific references which use this antibody for research. Gla is the abbreviation for the amino acid gamma carboxyglutamic acid, and this amino acid is derived from glutamic acid, which is abbreviated as GLU, by a process called gamma carboxylation, and this is a post-translational modification. In other words, this happens after the protein has been synthesized, and then some GLU domains are converted to Gla by gamma carboxylation. Gamma carboxylation is mediated by gamma-glutamyl carboxylase, a resident enzyme in the endoplasmic reticulum, and very important, this post-translational modification step is vitamin K dependent. A Gla domain is defined as a stretch of about 47 amino acids in which 9 to 12 GLU residues have been converted to Gla. Here is a comparison of the structure of GLU and Gla, glutamic acid and gamma carboxyglutamic acid. You see here glutamic acid with one carboxy group and Gla with two carboxy groups in the gamma position of the amino acid structure. Here you see a schematic description of the gamma carboxylation process, which leads to the formation of a Gla domain. You have the original protein stretch with GLU domains, and then the gamma glutamyl carboxylase, which operates in a vitamin K dependent fashion, converts these GLUs into Glas. Very important, Gla domains in a protein function as ligands for calcium. And calcium binding to the Gla domains causes a conformational transition that is crucial for the biological activity of proteins with Gla domains. And these biological activities dependent on this conformational transition induced by calcium are mostly interactions with cell membranes or interactions with other proteins. And of course, several important blood coagulation factors contain Gla domains and need this for proper biological activity. Here a schematic description. The Gla domains cause a conformational change in the presence of calcium. Gla domain, calcium binding, and then this transition, folding of the protein in a certain manner, which is important for proper biological activity. Several proteins with Gla domains are known. Of course, the coagulation factors, prothrombin, factor 7, factor 9, factor 10, protein C, protein S, and others. Certain bone proteins, like periostine, osteocalcin, matrix Gla protein, and certain snake venoms, like the conotoxins or factor 10-like proteins. The anti-Gla domain monoclonal antibody M3b was developed as a tool for research on Gla domain containing proteins. Despite ubiquitous expression of the gamma carboxylation machinery in mammalian tissues, only about 12 Gla containing proteins have been identified in humans so far. So there are possibly many other proteins which have not been discovered, and an antibody reacting with Gla domains would clearly be helpful in identifying such proteins. And therefore, the monoclonal antibody M3b was developed as a tool for identification and purification of Gla-containing proteins in human and non-human biological sources. And this was done by the research group of Brown and colleagues under the guidance of Johann Stanflow. Original publication on the antibody is in the Journal of Biological Chemistry in 2000. Shown here, the title: Identification and Purification of Vitamin K-dependent proteins and peptides with monoclonal antibodies specific for gamma-carboxyglutamyl Gla residues. Some experimental data is shown here. From this publication, you see here a Western blot, certain Gla-containing coagulation factors, prothrombin, prothrombin fragment 1, etc. And you see the reactivity of the antibody. And very important, decarboxylation destroys the antigenic epitope. You see here human prothrombin fragment 1. It's reactive. After decarboxylation, this reactivity is lost. Also from the publication of Brown and colleagues. Very important, the antibody M3b does not bind to Gla-domains in the presence of calcium. Shown in this experiment, you see here the reactivity of the antibody with of different antibodies which were generated series of experiments. Here you see the antibody M3b, and here is shown the reactivity with prothrombin, and you see that M3b reacts with prothrombin in the absence of calcium but in the presence of a other salt to correct for the ionic strength. Sodium chloride, see reactivity. In the presence of calcium, no reactivity. Here a control reactivity of a control antibody. Taken together, M3b does not bind to Gla-domains in the presence of calcium. Schematic description here. If there is no calcium present, there is no conformational folding of the protein. M3b does react. In the presence of calcium, folding, M3b does not react. No binding in the presence of calcium. And this behavior, of course, can be used for immunoaffinity purification of proteins with Gla-domains from complex mixtures. And here you see an experiment also from the publication of Brown and colleagues, where they have immunoaffinity purified tiger snake factor 10A-like protein from the crude snake venom. And what they basically did, they loaded a column with immobilized M3b with the snake venom. Then they washed the column, and then they eluted the column just by passing through a calcium containing elution buffer. And here you see the result of the immunoaffinity purification with the factor 10A-like protein from snake venom, which is a Gla-domain containing protein, being eluted from the column. What are the ADI products? There's the product number 3570, which is the purified monoclonal antibody against the Gla residues. It can be used for Western blotting in particular. And there is ADI product 3570MX, which is matrix-bound monoclonal antibody M3b, which can be used for immunoaffinity purification of Gla-containing proteins, as just shown in the last slide. More product information, including order information, is available at American Diagnostica's websites, americandiagnostica.com or americandiagnostica.de. Now I will proceed with some selected publications in which the antibody M3b was used. There is a publication by Stenberg and colleagues in 2001 in which they identified a functional prothrombin gene product in human kidney cells. This was the conclusion. This study, which used the antibody M3b, demonstrated for the first time the de novo synthesis of a full-length gamma carboxylated and functional prothrombin gene product by human kidney cells. There is a publication by Hall and colleagues in 2002 in which they isolated a Gla-containing protein from certain retinal cells, and they could show that Gas6, which is the protein they identified, requires gamma carboxylation in the vitamin K dependent reaction for functional activity. Coutu and colleagues, 2008, they could show that periostin, a member of a novel family of vitamin K dependent proteins, is expressed by mesenchymal stromal cells. Here the conclusion: the most abundant Gla-containing protein secreted by mesenchymal stromal cells was identified as periostin, a previously unrecognized gamma carboxylated protein. Brown and colleagues in 2002, they used the antibody for characterization of vitamin K dependent proteins in snake venoms, and again this was an example for using the antibody for immunochemical detection of gamma carboxylated peptides. I hope this information was helpful, and I thank you very much for your attention.